We usually accept cognitive decline, cardiovascular weakness, and slowing metabolism as the inevitable wear and tear of getting older. The standard medical assumption has long been that our biological systems simply degrade over time. But an extensive body of clinical trials conducted between 2017 and 2025 provides concrete counter-evidence. Researchers isolated a specific class of bioactive pigments called anthocyanins, the natural compounds that give certain fruits their deep red, blue, and purple colors. This split-screen timeline illustrates what researchers now call the blueberry effect. On the left, regular consumption delays cognitive decline by up to 2.5 years. On the right, clinical data shows a simultaneous 25% drop in cardiovascular mortality. By identifying the specific cellular pathways these pigments activate, we can actively intervene in our own aging process. Modest, regular intake, as little as 1/3 of a cup of a specific food, triggers these exact biological mechanisms. Biological aging is a flexible process. Specific, naturally occurring compounds can intercept and slow the cellular damage we previously thought was unstoppable. Clinical MRI studies reveal that anthocyanins directly increase blood flow and activation in the brain. In elderly populations, chronic supplementation improves episodic memory and task-switching accuracy. In one specific 6-month trial, older adults saw their processing speeds restored to the baseline levels of healthy reference groups. On an epidemiological scale, tracking over 150,000 individuals shows that high berry intake correlates directly with a lower risk of developing Parkinson's disease, while mitigating the long-term metabolic risk factors associated with Alzheimer's. This diagram shows 4 mechanisms anthocyanins trigger inside brain cells. First, potent antioxidant activity neutralizes free radicals, preventing oxidative stress. Second and third, they suppress neuroinflammation and regulate autophagy to clear damaged proteins. Fourth, reducing insulin resistance optimizes the brain's metabolic environment. Anthocyanins actively rebuild and maintain the structural integrity of the brain's neural network. The protection extends outward from the brain into the cardiovascular system. Clinical trials show anthocyanin supplementation increases flow-mediated dilation, a direct measure of how well blood vessels can relax and expand, by nearly 1%. This animated cross-section shows blood vessels physically widening, leading to the data in the descending bar chart. Better vascular function translates to a measurable drop in systolic blood pressure. Longitudinal twin studies also show this intake directly reduces total visceral body fat. Less visceral fat and a healthier lipid profile stabilize your metabolism. Regular consumption, even as infrequently as a few times a month, is associated with a 23% to 26% reduced risk of developing type 2 diabetes, because anthocyanins help normalize glucose metabolism and reduce fasting insulin. These molecules also cross the blood-retinal barrier. In patients with normal tension glaucoma, they stabilize visual field loss and improve ocular blood flow, while short-term studies show they actively reduce general eye fatigue and inflammation. Introducing this single class of compound initiates a compounding wave of protection, defending the brain, protecting the heart, stabilizing the metabolism, and supporting the eyes simultaneously. To get these exact biological results, you need an actionable protocol, and that starts with evaluating which dietary sources actually contain the highest concentrations of anthocyanins. This descending bar chart reveals the clear winner. Wild low-bush blueberries yield 487 milligrams of anthocyanins per 100 grams of fruit. They easily outperform cultivated high-bush blueberries at 387 milligrams, blackberries at 245, and completely eclipse strawberries, which carry a mere 21 milligrams. Once you have the right source, Rule 1 of processing is simple. Cold is your friend. This line graph charts the stability of the compounds over 10 months. The high blue line shows that rapid freezing at -18 degrees Celsius preserves the molecules entirely, resulting in only a 12% loss over nearly a year. Rule 2. Heat is the enemy. Thermal processing, like pasteurizing juices or making jams, destroys the target molecules through oxidation. Storing those products at room temperature accelerates the degradation, plummeting the half-life of the anthocyanins from over 100 days down to just 12. The clinical trials confirm that you only need about 50 milligrams of anthocyanins daily to see significant health benefits. You can hit that target by eating approximately 1/3 of a cup of frozen wild blueberries. Earning these dialogical benefits requires consistency. Over 50% of ingested anthocyanins remain in the body 48 hours after consumption. A small daily habit ensures a constant supply to your cells. Long-term biological resilience is an active choice. Adding a handful of frozen wild blueberries to your daily routine provides a proven, accessible defense against the mechanics of aging.